alluding to before, look how much more secondary xylem there is than secondary phloem. Secondary phloem is not as important as moving the sugar water down as it is water going up. You need a lot more water going up, and so not only does wood, sapped wood, provide a lot of water goes in, but it also provides a lot of strength. Okay, something to think about. And so now look at this. We're going further down. You can see now this tree is getting thicker towards the bottom, and the reason it's getting thicker, it's growing laterally, so girth. The reason it's getting thicker is now look what you've got. The vascular cambium is put down not only 1 year. Well, this is still 1 year, but it's the late wood and the early wood. But you see, here's the sapped wood, here's the secondary phloem, here's the secondary xylem. And as the years go on, this layer right here, let me emphasize it, this layer right here will eventually make the ring. So it'll be 1 year, and then the next year, and the next year. And so in temperate regions, the secondary growth in a perennial plant ceases in the winter. Well, that means that, you know, in an area where the leaves are deciduous, in other words, they're dropping their leaves, obviously there's not going to be a lot of growth occurring at that time. You know, maybe in the tropics, where the leaves are there perpetually, they're always, perpetually, I have to say, that things are a little bit different. And so the thickness of the cells provides physical support. That's what I wanted to say. And so you get this sort of dormancy, and then you get early wood production, late wood production, and that's what results in the growth of the annual range. So as you can see here, this is 3 years. So 1, 2, and 3. Here's your vascular cambium. This is all secondary xylem right in here, secondary xylem. This is the original. This is secondary xylem. This is then secondary phloem, and a little bit of primary phloem out there, and then cork cambium, and then cork. So this is a picture showing like fire scars right here, and you can sort of date these. But look over here. Even without a microscope, you can see that these lines represent the years of wood, which are the years of secondary xylem. You now know. And so cork cambium is a meristematic tissue that forms on the outermost part of the tree, and it results in the formation of these cork cells. Cork's really important. It has a lot of this waxy material. You're familiar with what cork feels like and the texture of cork. It has this waxy lipid material which prevents water loss, but it also is a barrier against physical damage and also pathogens, and also fire. Extremely thick. It can be up to like a foot in diameter. It's pretty impenetrable, and so that's a clear adaptation to fire. And so this is a really nice diagram here. This shows inside, okay, let's look at the green again. This is the vascular cambium. The vascular cambium is a lateral meristem, and so is the cork cambium. The vascular cambium is producing secondary xylem, and it's also producing secondary phloem. The cork cambium is producing cork. So this is considered to be the sapwood and the heartwood. I'll come back to the heartwood in a moment. Bark is a combination of periderm and phloem. So you can't remove the bark of a tree without killing it, because you need that phloem. And so, again, here's a diagram. This is labeled. It's not under the microscope. Again, here is secondary xylem, right here, secondary xylem. Here's the late wood and the early wood, meaning dry and rainy seasons. Here's the secondary phloem. Here's the vascular cambium. And there is the cork cambium, and it's hard to see it, but these are the cells that are dividing, producing the cork. And so here's another picture. Vascular cambium, secondary xylem, secondary phloem. And so bark refers to the secondary phloem, the cork cambium, and the cork. So you need to have that in order for the plant to be able to survive. And so here's another cross-section. Different pictures will help. Here's the heartwood. It's a little bit darker in coloration. The sapwood. Here's the vascular cambium, which makes the xylem and the phloem, and the cork cambium, which makes the cork. And so look at this picture here. I'm going to talk a little bit about the heartwood right here. You can notice how this area is a little darker in color. It's still xylem. There's no doubt about it. There's xylem, and then this lighter wood right here is also xylem. So this is secondary xylem. So you're like, well, where's the vascular cambium? It's really towards the outer part of the plant, from the tree. And there's a lot happening here. We've been looking at slides under the microscope. And so this is all secondary. So why are the darks on the inside? The heartwood no longer conducts water. So the sapwood, I just want to emphasize this. This is rather important. So the sapwood is here, this lighter color, sap. And then this darker area right here is considered to be the heart of the tree, the heartwood. Now, it no longer conducts water. What's good at it? It's like, well, it supplies the tree with tremendous stability and strength on the inside. And that's because its xylem, it gets all lignified. In other words, do you remember that substance that was sort of this not a carbohydrate, but an organic molecule with a lot of alcohol groups? And it helps to sort of darken the wood, and it helps, and these resins help the core of the tree, not only for strength, but also protect it from other, wood-boring insects and fungi that are trying to hurt it. So the heartwood is really important for the life of the tree, but it doesn't conduct water so much. Check this out. The heartwood doesn't conduct water. I was mentioning that before, but the sapwood does. And so that is your living. Not living because it's dead, and it's dead. But that's what's conducting water up the tree. It's no longer alive, but it's really important sapwood. And so here's a picture of the hardwood. In the center here, here's your sapwood. So all of that secondary xylem. Here are the rays that are moving laterally out here. Here is the vascular cambium, which is producing the phloem, which is rather thin. So this is a maple tree, and you wanted to get some maple syrup to come out of this. All you have to do is just go just below the bark, and you'll penetrate into this thin layer called the phloem. And that's what will produce the sort of the sugar water, which is what we like about maple syrup. And then all of this is the secondary xylem. It's pretty cool. And so I thought I'd show you this picture. This is a giant sequoia redwood tree, of course. And this is in Yosemite National Park. It's a picture a long time ago. You can tell by the car here. This is in an area called Verona. And this is a kind of, in some ways, tragic, in some ways, interesting historically, because the early people that were sort of hanging out in the redwood trees thought it would be clever to cut a tunnel through these trees. They're so wide in diameter that it will allow, like, horses and carriages to go through, and then eventually automobiles are able to go through it. So it's quite a big tourist attraction. People came around the world to check this tunnel tree out. You're like, well, is the tree going to die for putting a big hole in it this way? This is a good question. Most of the damage done to the tree, by the way, this would not be allowed today. We know this isn't happening. But the inside of the tree, mainly the damage is the hardwood on the inside. And that doesn't affect the movement of water up this redwood tree, which is mainly in the sapwood, which is towards the outer part. So the tree can live for many years, even after this mutilization. And so as we sort of progress, like just a little review, if you don't mind, from on the very top of the plant, we got this apical meristem, right, at the very tip of the tree. And that's primary growth, so that's going up towards the sky. And then we got these primary meristems, protoderm, which becomes the epidermis, the procambium, which becomes the vascular tissue, and then the ground, which becomes the cortex of the stem, or the pit of the inside. But eventually, the lateral meristems, which there are two, vascular cambium and core cambium, the vascular cambium will eventually produce the secondary xylem towards the inside and the secondary phloem, and then the core cambium produces the cork, which is then considered periderm. That's kind of an interesting flow right there. And so the two lateral meristems, vascular produces the secondary xylem and phloem, and core cambium produces the bark, ultimately the cork. But there's also secondary growth in roots. I'm not sure if you realize this, that even within the roots can become rather thick as well. So what's interesting is that though the root can develop bark on the outside of it, that isn't where water is being absorbed. And so deep in the soil is where the young roots are, which are very thin. And that's where water absorption and mineral absorption takes place. That is, again, if you're a redwood tree, and then your roots are a little bit more closer to the surface, and they grow more laterally than they do deep. And so secondary growth in roots, check this out. It's almost like an extension of the stem itself, but it's thick. They're thick and they're impenetrable. And so this helps to provide further support of the tree. It helps to anchor the plant down. It helps to transport water, but it doesn't absorb water. And so finally, you can see here, check this out. This one has so much bark on it that it even has some of the roots that actually have lichen growing on it as well, which is kind of cool. So over the years, the roots can become woodier and tough bark develops. But this is not where water is being absorbed. That's happening much deeper down below. So I hope you enjoyed this video on secondary growth in plants and the formation of wood. Thanks for watching. Everyone, it's Mr. Cinti, and I'd like to talk to you a little bit about bacteria. And in particular, I'd like to talk to you about how bacteria transfer their genetic material from one bacteria to another. And there's 3 ways to do that. Before we start getting into the detail of the types of genetic recombination of bacteria, let's talk about a little bit of why this is an important idea for bacteria to have, the ability to do this. And so let's jump right into this conversation. And so let's say, for example, right out of the gate, one of the most important things to consider about bacteria is that they have such a short generation span, short generation time. So in optimal conditions, we're talking about 37 degrees, plenty of food. We're talking about 30 to 40 minutes, a bacteria will be able to grow and divide into 2 daughter cells, which are clones. And so this short generation time and the ability for them to mutate often allows them to adapt to changing environments really well. And so this is how they do it. This is how they're most successful, is that they're extremely adaptable bacteria. I mean, they can take anything. You can grow, change temperatures, you can change different environmental conditions, you can throw antibiotics at them, and some of them are going to make it through. And it's because of mutation rate and short generation time and their ability to swap genetic material. So in a true sense, in an evolutionary sense, their ability to adapt via natural selection is because they're very good at adjusting to changes in their environment. And so let's take a look at this. It all comes down to the genetic material. Most of the DNA inside of a bacteria is in this one big chromosome, which is clustered together in the center called the nucleoid. And there's, you might think there's a lot of genes on a bacteria. There's around 4,000 or so genes in a typical bacteria. We have approximately 23,000 genes. And so it's quite a few more. But then again, they have a lot more than a typical virus. So what's interesting, in addition to that chromosome of DNA, they also have these circular sort of rubber band small quantities of DNA in the cytoplasm called plasmids. They're sort of like rubber bands if you're wearing braces that would hold your teeth together. And so these rubber bands or these plasmids of DNA are small rings of DNA, and they often carry just a few genes. Some are carried for resistance to antibiotics, so it's kind of cool that if they have that, cool if it's a bacteria. Some of them even have genes on them for producing the pili, which are those finger-like extensions on bacteria that allow them to sexually reproduce or conjugate with other bacteria. So those are called F plasmids if they have that for fertility, and R plasmids if they carry resistant genes. In a future conversation, we'll talk about how plasmids are very useful to biotechnology. We've been able to pull plasmids out of bacteria and cut them with restriction enzymes and include foreign DNA into them and use them as a vector to transfer genetic material to one bacteria and then clone them up so we can amplify bacteria using plasmids. So plasmids are really, really, really important in biotechnology, and they're really important in the bacteria. They just have a few genes, but they're crucial ones. So this may be a review, but when a bacteria, remember I said it only lives around 30 minutes before it replicates, and so it's getting busy. There's one origin of replication site, DNA polymerase comes down, attaches, and starts replicating. Here's a replication fork here, and it copies its DNA into two. You may recall that once the DNA is replicated, it simply attaches it to the membrane, and then when those membranes increase in size, those copies of the DNA move along, and then the cells get to divide through binary fission. It doesn't have to do the whole mitotic thing as more easily as it doesn't need spindle fibers. It's really simple, gets the job done. It's very efficient. So bacteria, if anything, are very prolific. They reproduce, and since there's a lot of them, their growth rate is rather rapid. So it's like a lot of them, and they grow really quickly. So there's the exponential growth increase can be really dramatic. So you can see here in just a few minutes, they can increase quite a few, and then if you give them like a half day, they can go even further. And so our whole body's covered with these, and a lot of, this is not necessarily a great thought, but a lot of our feces contains bacteria in there. And so, as I mentioned before, they reproduce through binary fission, and one of the things about this is that when they reproduce, their DNA polymerase, so quick, is not very good at proofreading. There's not a lot of bacteria that have proofreading DNA polymerases. You're like, oh, it's not as good. Now, actually, this is the key to the bacterial success. The fact that they make spontaneous mutations, in other words, errors in replication, allows for a tremendous diversity of bacteria, or else they would simply be clones of one another. And so, check this out. This means that if a spontaneous mutation rate is 1 in every 10 to the 7 mutations per gene per cell division, that means 2,000 bacteria in our intestine are going to have a mutation per day. That's a way in which to generate diversity. And so, this is really, really important. So, new mutations, though individually rare, overall, can have a significant impact on genetic diversity, especially when the reproductive rates are high and it's a short generation span. And so, bacteria use mutation and a rapid increase in their numbers through short generation time to be adapted to their changing environments. And so, to contrast that, we as eukaryotic organisms, we do have mutations and we do have new alleles, but we don't have such a quick generation turnaround. And so, how we increase diversity in the next generation is through sexual reproduction. So we use meiosis to sort of swap genes in our own cells in the production of gametes, and then sexual reproduction in general shuffles the deck between the DNA. And so sexual recombination is the way eukaryotes do it, and mutation and high reproductive rates are the way bacteria do it. And so let's talk about the 3 ways in which genetic material can be recombined in bacteria. It's really important because if there is a mutation, maybe they want to share it with other bacteria, and this is the way bacteria can recombine DNA with other bacteria. You might not think it's possible because let me sort of state the situation here. Here's one bacteria, and here's a separate one. So what I'm talking about is, like normally, the DNA will replicate itself into 2, and then the cell will divide, and then the cell and the cell are complete clones from this one cell. And so, that's great. So if there's a mutation, maybe there's some change in the population. But what I'm talking about is, like if you have a bacteria here, and I'm talking about a different bacteria here, how can DNA exchange from one bacteria to another? And there's 3 ways in which it can take place. One is your good old transformation. In other words, if you remember this, like the cell can break open and some of the genetic material simply just come out, like this. I was able to draw it. Let's go a different color here. Say the genetic material were to come out, maybe if I did a pen, it would be better. So DNA can come out here in between cells, and then it can be incorporated, and then it causes this cell to become transformed. So that's one way. Transduction involves a bacteriophage. And when a bacteria, I'll go over this in detail in a moment, when a bacteriophage injects its DNA and it starts taking over the cell, sometimes by accident, it will actually incorporate some of the bacterial DNA in its capsid. And so when the cell lyses, it goes over here, and when it infects a different cell, what it's doing is actually inserting, like a syringe, foreign DNA from a different bacterium. That's called transduction. And in conjugation, if you watch the previous video on bacteria, you kind of know where this is going. Some bacteria have these extensions called pili, and the DNA can actually travel through these extensions from one to another. So this is sort of like sexual reproduction in bacteria conjugation. So there's 3 ways in which bacteria can genetically recombine their DNA with other bacteria to produce new strains. We'll look at some examples of why that might be important too. Here's an example of that. Say, for example, you're growing E. coli or any other bacteria in a test tube. You're going to have some sort of nutrient in here, some sort of medium. Let's say it's minimal medium. And there's a mutant strain that, you see, notice here, it's tryptophan negative, meaning that it cannot produce tryptophan. And if it cannot produce tryptophan, sorry, you're out of luck. If it's not the food, you're not going to be able to live. So, no comments. So sorry. And then over here, look at this mutant. This one is arginine negative, meaning that it cannot grow in minimal media. Now, of course, if you gave it arginine, it would grow. And over here, if you gave it tryptophan, it would grow. But check this out. If you take the 2 bacteria and you put them in the same medium, and they're able to grow in here, sometimes there's a genetic exchange of DNA. And so there's some colonies that will actually pick up a little DNA from the other bacteria. And as a result, this one, when it grows on minimal media, it is able to grow because, interestingly enough, it picked up sort of the goodness of this one and the goodness of that one. And so therefore, it's sort of like a nice couple that's in a symbiotic relationship of mutualism. This one provides a little something good and this one a little something good. So there's a great example of why recombining DNA with other bacteria can be useful. And so let me refresh you with transformation. We've been through this a couple times, but this is Fred Griffith's classic experiment when he's killing mice over here. And do you remember the dead heat-killed S lost some of its DNA and it got incorporated into the R, which then transforms into the S, which killed the bacteria. And so that's an example of transformation. And it's simply the movement of some DNA or some alleles are replaced by the foreign transfer of DNA, and therefore it's incorporated. So the cell is now able to produce a protein, produce resistance, produce a protein capsid, a sugar capsule. So it's able to do things that it's never been able to do. It's been transformed. It's a basic, in other words, just diffusion of nucleic acid from one bacteria to another. What's interesting about that is that there's a few bacteria that are particularly good at that. They have proteins that it will be able to take up naked DNA. But as it turns out, E. coli, which is one of the more highly studied model prokaryotes, it's funny, but it doesn't do that really well. And so we have to make E. coli receptive to taking these DNA. So we learned that if we treat E. coli and grow E. coli prior to the transformation with calcium ions, calcium chlorides, rather simple salts, we've learned that that makes them more receptive to taking up DNA. We call that making them confident. So we can make E. coli confident by giving them some calcium chloride. And so what we do is we grow the E. coli with some calcium chloride, and what we think is happening is that the calcium chloride, when it's in solution, disassociates into calcium ions, and we believe that those cations adhere to the phosphate groups along the DNA chain, and therefore make it a little bit more neutral, therefore maybe make it a little bit easier for it to be taken in by E. coli. And then this other way in which bacteria can transform genetic material is transduction, and that's by using a phage, sort of like as a mosquito vector. And so there's going to be 2 kinds of transductions, general and specific. I'll talk about general first. It's when, let me go over here and sort of show you, here's a typical phage that's injecting its DNA into a bacteria. And so let's go here. What I want to do is show you on this board here something that's going on. So here's E. coli. Here is the phage. So the phage is attaching here. And what it does is it injects its DNA into the host. And so when it does that, if you recall, one of the enzymes that the phage carries is an enzyme that sort of dices up the nucleic acid of the bacteria. And that way it doesn't have to compete so much with that. So when it's doing that, another thing that's simultaneously happening is that it's making more viral capsids. So I'll just do this, like this, making more viruses. And so what's supposed to happen is the phages do this. They put their own DNA once it's been replicated here, but sometimes by mistake, they'll put a little piece of bacterial genetic material inside the protein capsule. And so when the virus ends up destroying the bacteria and the cell lyses, and all these little phages come out, what can happen is, for example, this one can go over here and infect another bacteria. And when it does that, it's not going to destroy the bacteria, but in fact, we can sort of see this. It infects it by inserting a little piece of this bacteria's DNA. So this is called generalized transduction because, who's to say if that's going to even happen? And who's going to say what gene is going to get incorporated into the phage? So it's totally random, so it's generalized. But the truth is, this bacteria is going to have a novel ability that it never had before because the virus helped to genetically recombine these 2 bacteria's genomes. And so let me talk a little bit about specific transduction right now. This is really cool. Like here's E. coli. Here again is the phage that's attaching to it like this. So when the phage DNA goes in, here's the bacterial chromosome like this. And so do you recall there's a phage called lambda-phage? And what happens is when its DNA is inserted into the bacteria, its DNA becomes incorporated into the host DNA, that. And so it becomes a prophage. And so this is known as a temperate virus. The DNA lays latent. It can be copied many times. And then what's interesting is something causes the DNA, the prophage, to release itself. And when it releases itself from the bacterial chromosome in future generations, sometimes when it excides from the chromosome, it actually pulls away a little bit of the bacterial DNA with it. So here it is, BGBB, breaking up like this. And so what's interesting is if I were to attach letters to this, this is G and A, B, C, E, D, E, F, G, H. I get the point. So the reason this is called specialized transduction is that it's not random which piece of DNA is incorporated into the phage. It's the one that's adjacent to the prophage, which is often the one that's going to get incorporated. So that's clear. And then what happens is this is what happens in the next phage. So a little piece of DNA is able to move over to the next bacteria. Let's go over here, back to our slideshow if we can. So here is an actual picture of a phage infecting your bacteria. So in specialized transduction, it involves a temperate phage. The prophage inserts itself into the bacterial chromosome. And as you can see over here, here's the prophage. It's kind of small. But when the prophage removes itself from the chromosome, it's carrying a little piece of DNA with it from the bacteria, as you can see here. It's A+. And so when it infects a future bacteria, it brings that A+ along. And then over here in general, comes in, splices up the DNA, and then just randomly picks up a piece. So those are the two side by side. And then finally, the third way in which bacteria can transmit or genetically recombine their DNA with another bacteria is if one has pili. So you see this one has a lot of pili on it. Do you remember it? It's the pili. This is kind of slightly humorous. The bacteria that have pili are considered to be male because they have these bridges that will allow DNA to exchange across. And so here is a picture of bacteria reproducing or conjugating. And so the male one is sending DNA over to the female, and the female receives the DNA through the sex pillars between the bridge between the two bacteria. And so there you have it, the three ways in which genetic material is exchanged between bacteria. So high mutation rate, high reproductive rate, short generation time, transformation, transduction, and conjugation make for bacteria to be extremely adaptable and therefore, no doubt, the most successful organisms on this Earth has ever seen in 3.5 billion years. So hopefully that was helpful, and you enjoyed it as well. Thanks for watching.
Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about how the genetic code was cracked. When I say the genetic code, I mean the sequence of nitrogen bases. It's how it was figured out in terms of what the nitrogen bases meant in terms of a polypeptide sequence of a protein. Now, on surface, when you talk about that, it's like, well, is that significant? Well, of course it's significant. It's cracking the genetic code. We're talking about one of the great discoveries of all time in biology. And so what I want to do is get into this conversation in terms of a story, and I want to describe some of the experiments that led to cracking the genetic code. Because I'm going under the assumption that we are familiar with the fact that the genetic code is located on messenger RNA, and that's translated by ribosomes in the cytoplasm of both prokaryotes and eukaryotes. And going under the assumption you know that. And I'm going under the assumption that you know that it's a three-letter codon that corresponds to each of the 20 amino acids. But I want to talk to you sort of like, again, a story of some of the science that's involved in cracking the code, because the reason I want to do this, and I want to introduce you to the Nobel Prize-winning scientist Marshall Nuremberg, who is instrumental in cracking the genetic code, is because I think at this stage in my career in biology, I find it more valuable to get into sort of the head of the scientists and sort of the methodology and research design, more than it is the content of the material. I apologize for that. I'll update that later. And so it's more important that you understand that the information found in textbooks or on the internet is really the result and the conclusions of some of these great experiments. And so this is, I'm not sure if, there's many great experiments, but this is certainly one of them, and I hope I can give it justice. And so Marshall Nuremberg has this dilemma. In other words, when you look at RNA, and this is a single-stranded molecule of RNA polymer, and you notice the nitrogen base is sticking out, the question is, how is the genetic code interpreted? So in other words, like the nitrogen base sequence is the genetic code, but like what is the code? In other words, I mean, we like to think that maybe it's a single